My name is Parshva Shah, and I'm a fourth-year medical student. My James Scholar project is titled Studying Neuromuscular Defects in Spinal Muscular Atrophy Using Patient-Specific Induced Pluripotent Stem Cells. I want to thank Dr. Chen and my faculty advisor, Dr. Lee, for all their assistance with this project. We have no competing or financial interests to declare. Background. Spinal muscular atrophy is the leading inherited cause of infant and toddler mortality. It is an autosomal recessive disease, and it's caused by a homozygous loss or mutation of the SMN1 gene. This results in decreased expression of the SMN protein. The disease itself is a lower motor neuron disease characterized by degeneration of anterior horn cells within the spinal cord, and it remains the most frequent genetic cause of infant mortality. Early pathological changes are detectable at the neuromuscular junction. Specifically, the clustering of acetylcholine receptors is impaired. Another early pathological change that's noticed is the reduced number of mitochondria, mitochondrial area, and mitochondrial transport. This defect has been rescued using N-acetylcysteine, an antioxidant that mitigates mitochondrial stress. The mechanisms involved with neuromuscular junction development and the underlying pathogenesis remain unclear. Recent studies have pointed to the involvement of mitochondrial dysfunction in SMA using animal and cell models, but its role on neuromuscular synaptic defects is still unknown. Humans are unique in that they also have two SMN genes rather than just one, and thus it's important to generate human SMA models to study the disease. One way to do this is by using stem cells. The goal of this project was twofold. First, to look at and identify the neuromuscular junction defects in SMA iPSC-derived neuron and muscle co-cultures, and then to test whether these synaptic defects can be rescued using factors like NAC. For methods, we first took our iPSCs and took them through the differentiation process. So we took our pluripotent stem cells, took them to the neuroectoderm stage, and then neural progenitors, ultimately forming motor neurons. And finally, these motor neurons were co-cultured with human muscle cells to develop neuromuscular junction. To help assess this process, we also used RT-PCR and qPCR, looking at various gene marker levels, such as GAPDH, OCT4, SOX2, PAX6, HB9, and OLIG2. Finally, we introduced N-acetylcysteine treatment. So the motor neurons co-cultured with muscle cells were treated with or without NAC. We created 3 cell lines, GM1, which was our normal control, SMA232, and SMA3813. Finally, we also did immunostaining, where we stained for anti-BTX and anti-CHAT. And we calculated the number of NMJ puncta formed. Statistical analysis was done with a two-way student's t-test, and statistical significance was deemed as a p of less than 0.05. Results. We first looked at our cell differentiation and co-culture with the muscle cells. At day 0, we had pluripotent stem cells. By day 14, we had generated neuroectoderm, and by day 30, we had neural progenitors. Finally, if you look at the right picture, you'll see this is our co-culture with the muscle cells. So here we have our motor neurons and muscle cells forming connections and developing neuromuscular junction. Next, we looked at our GAPDH loading control. This is a gel electrophoresis showing the DNA expression levels of GAPDH within each of our cell cultures. This was important to do because we wanted to show that the DNA expression levels within each of the cultures was roughly equal, and that any deficits that were noted were due to the disease process itself. Next, we looked at the qPCR quantification of OCT4, PAX6, SOX2, HB9, and OLIG2 cell markers at day 0, day 14, and day 30. With OCT4, we see that at day 30, there is significantly decreased expression relative to day 0. Specifically, GM1 saw an 18-fold decrease, SMA232 saw a 9-fold decrease, and SMA3813 saw a 6-fold decrease. SOX2 levels steadily increased by day 30 relative to day 0. We also see that PAX6 levels spike at day 14, followed by subsequent decline by day 30. And this correlates with the neuroectoderm stage of differentiation. Finally, looking at HB9 and OLIG2, we see that those levels increased by day 30 relative to day 0. Specifically, with HB9, we see in GM1 a 219-fold increase. In SMA232, we see an 870-fold increase, and SMA3813 noted a 174-fold increase. Within OLIG2, we also see similar increases, with GM1 showing a 17-fold increase, SMA232 noting a 30-fold increase, and SMA3813 noting a 33-fold increase. Next, we looked at our immunostaining of BTX and CHAT. Here we are looking at GM1 without NAC treatment and GM1 with NAC treatment. This is our non-diseased model, and so we see relatively strong staining of both CHAT and BTX. Next, we're looking at SMA232 vehicle, which is without the NAC treatment, and SMA232 with NAC treatment. Here we see that SMA232 with NAC treatment has stronger BTX staining compared to its non-treated counterpart. We see the same results in the SMA3813 model, where the NAC treatment has stronger BTX staining compared to the SMA3813 vehicle lacking the NAC treatment. Finally, we quantified the BTX to CHAT staining ratio. Here we see that there is a 53% decrease in SMA232 minus NAC relative to GM1 minus NAC, p-value of 0.02, and we also see a 60% decrease in SMA3813 minus NAC relative to GM1 minus NAC with a p-value of 0.002. Next, the other thing that we see here is that in both the SMA232 and SMA3813 models, the positive NAC groups or the NAC treated groups have increased BTX to CHAT staining ratio relative to their non-treated counterparts. We see a 91% increase in SMA232 with NAC relative to its non-treated counterpart, p-value of 0.01, and we see an 80% increase in SMA3813 with NAC relative to SMA3813 minus NAC with a p-value of 0.04. Discussion. So overall, we feel that we were pretty successful in differentiating our stem cells into normal and spinal muscular atrophy diseased motor neurons. We are able to say this because there was a significant decrease in the mRNA expression of the pluripotent stem cell marker OCT4 during neural differentiation, and PAC6 levels increased at day 40 with a subsequent decline by day 30, which was consistent with our neuroectoderm stage of differentiation. And finally, the expression levels of HB9 and OLIG2, both of which are the motor neuron markers, significantly increased by the end of the differentiation process. Together, this helps support our notion that we were able to successfully go through the differentiation process. Next, looking at our NMJ model and the rescue with NAC, we can say that SMN deficiency impairs NMJ formation. Compared to the normal GM1 model, both the SMA232 and SMA3813 result in a statistically significant decrease in NMJ puncta formation. And also, we're able to say that this disease-related reduction in NMJ formation can be mitigated by the introduction or treatment with NAC, as the SMA232 with NAC and the SMA3813 with NAC cultures both showed a statistically significant increase in their NMJ puncta formation compared to their non-NAC counterparts. In conclusion, this means that the regulation of mitochondrial function could rescue the defects that are observed at the neuromuscular junction in SMA. Some limitations of our studies were that due to cell death, our total samples for the SMA232 with NAC was limited to 7, while the other control and treatment groups had 10 plus samples to contribute to the analysis. Another limitation is that we had an in vitro model, and so conditions present with an in vivo model could result in potentially different outcomes. However, the results do show a promising trend for the rescue of NMJ defects. We do still need further investigation to assess its clinical application. Future directions to take this study in could be, one, to investigate the effects of differing concentrations of NAC on neuromuscular junction formation. And the second one that I'm more interested in would be to investigate the effects of using an antisense oligonucleotide in conjunction with NAC treatment for SMA. In December of 2016, the FDA approved the use of antisense oligonucleotide drug to treat SMA. This drug works by upregulating the human SMN2 gene, leading to increased production of SMN protein. NAC offers a different level of intervention, and so the use of both of them together could result in a vastly improved response, potentially for patients with SMA. In conclusion, the deficiency of SMN impairs NMJ function in co-cultures of human stem cell-derived spinal motor neurons and human muscle cells. Our results also suggest that NAC and the introduction of NAC specifically can mitigate this disease-related reduction via the regulation of mitochondrial function. Overall, the results of the study provide insights into developing systems for testing therapeutic interventions aimed at preventing, delaying, or even curing SMA. These are my references. Thank you very much for taking the time to listen to my research project.